Hi guys, it's Mrs. Mancuso, Merm here, ready to start our DNA unit. So our DNA unit is called Molecular Genetics. The first video is going to be all about the experiments that helped us understand what DNA is and what it's responsible for. So we start off with this guy named Griffith back in 1928. You don't have to memorize dates or things like that. I just want to put it in perspective for you. So back in this time frame, he was actually working on trying to find a vaccine for pneumonia bacteria. So he ran an experiment with mice and he had two different strains, an R strain and an S strain. And pneumonia, again, is a type of bacteria. R stands for rough. So the rough strain actually had a rough edge around it, and your antibodies could attach right to that rough edge and kill it. So this did not make you sick. The R strain was fine, but the S strain, S stands for smooth, it had a really smooth coat around it and your antibodies couldn't lock onto it. So this is what you call a very virulent or deadly strain. So this strain kills because your antibodies can't attach to it. All right, so he ran an experiment with mice, and I'll show you the nice picture in a minute. But let me just show you what he did. So if he takes and he gives the mice the R strain, they live. No shocker there, because we just said the R is rough and your antibodies attached to it, so we would expect them to live. It's not virulent. Give them the S and they die. Again, no big shocker. We know that the S is smooth and they can't attach, so it is very virulent and it kills the mice. And then the next one, he heated the S and the mice live. No shocker there, because we know if you heat up the bacteria, you're going to denature it. And so by denaturing it, like they had known by 1928 that if you heat your food, you're going to kill the bacteria in it. So that was not a surprise. Here's the surprise. If you have heated S plus your regular R, you would expect the mice to live, right? And instead, they died. And he was like, what the heck is going on? The heated S isn't virulent because it's denatured, and the R didn't kill you before, so what is happening? So he called this, what must be happening, a transformation. And there must be some sort of transformation factor. There has to be something that is changing the R into S, but he didn't know what it was. And let me tell you that when they pour money into research, they want to put money into something that they believe could actually be it, so the research would be worth it. So back in this time, it's really a fight between, is it protein or is it DNA? And the money was going into the protein research, because think about what we know about the structure of a protein. We know it's very different, that there's a diversity of proteins, that there's 20 different amino acids, and they can come together in so many different ways, give you multiple different shapes and multiple different functions. But if you think about what we know about DNA, there's 4 bases, A's, T's, C's, and G's, and they just keep repeating. So what's the variety in that? How can it really be DNA that's so important? So that's why it was taking quite a while for research to happen. Our next scientist, oh, here's the pretty picture. So there's the R strain causing the mice to live, the S strain causing the mice to die. Heat up your smooth and the mice live, and then there's our shocker. The heated S and the R cause the mouse to die. All right, so here's our next experiment. Avery, McCarty, and McLeod, over 10 years later, what was the necessary ingredient to be the transforming factor in the experiment? They were trying to figure out what Griffith wasn't able to answer. What was it that was required? So they would remove one key ingredient at a time, take out the protein, take out the lipid, take out the carbs, take out the RNA, take out the DNA, till you see what causes mice to be treated with the heated S and the R to actually die. So I'm going to show you this in a little visual. So if this is my solution, so I have my heated S and my R, and I take out the proteins. All right, the mice, so if I have heated S and R, the mice die, and that is because the R's changed to S's. So S's were present in this solution. And then if I do it again, and again, I have my heated S and my R, and this time, this is a minus sign, so minus the proteins. This time, let's say I take out the carbs. They really tested it with everything. RNA, carbs, lipids, I'm just gonna show you some of them. Take out the carbs, the mice still die, and the R did change to S. So S were present in this. And then I'll show you a third one. They take the heated S plus the R, and this time they remove the DNA. Ooh, DNA. Don't know what I was doing there. All right. Take out the DNA, and the mice live, and there is no change. There is no S present. No S means that they're not going to die. So if you take out the DNA and your experiment doesn't work the way it did before, the DNA must have been the necessary ingredient to change the R's to S's. So this experiment really figured out that the transforming factor was DNA. So here's, again, I like to show you a little diagram that's a little neater than mine. So here's that original solution. They heated the S. So if you heat the S and add protein ACE, so something that will remove the proteins, very general term there. That's going to take out the protein, add the R cells, and you get S cells. So they did change. So protein must not be that key ingredient. Over here, add RNA ACE. That takes out all the RNA. You add R cells, and they still change into S. But over here, if you add DNA ACE, you're going to remove the DNA, and now you get no S cells. So the transforming factor to change a heated R into an S must be the DNA. And there it is, just again, what was required to do it. All right, Hershey and Chase. This is our next experiment, 1952. So they looked at what do bacteriophages. So bacteriophages are those funky looking, they look like weird little rocket ships. They are viruses. So this outer coat is a protein, and it has some sort of DNA or RNA inside of it. So these bacteriophages, viruses that infect bacteria, so phage means virus, whatever's in front of it tells you what they attack. So what do they pass on to bacteria cells? They used radioactive tags. So again, if the race or the understanding is between proteins versus DNA, I want to use tags to see where they end up. I like to think of this as they were chasing tags because it helps you remember the scientists. So Hershey and Chase were chasing tags. So the tags are going to radioactively label this virus. So the virus, we said, has two parts to it. It has the outer coat, which is protein, and it has the inner part, which is DNA. If they use a different tag for each one, they have to come up with something that is unique. What element is unique in a protein that you do not find in DNA? And that's sulfur. So they tagged the protein with sulfur. So I'm going to use a little yellow around it. So that is my radioactive protein. And then they used radioactive nitrogen to tag, oh, this should be an S, sulfur to tag the proteins. And then actually they couldn't use nitrogen. I just said nitrogen. Why can't they use nitrogen? Nitrogen is in proteins and it is in nucleic acid. So you have to use a radioactive tag that is unique. Phosphorus is unique to DNA. So use phosphorus to tag the DNA. So I'm going to tag this with a P. So all of this inner part will be tagged with the phosphorus. And then they let them do their work. So here's the bacteria, and they wanted to see what radioactive labels end up in the bacteria. So when the bacteriophages infect the bacteria, only phosphorus tags are detected. Sulfur tags were in the solution. So when they passed on, here is my new bacteria, which I really need to make in another color. So let me erase it, because I don't want to use purple. Purple is my tag. So I'm going to say, here is my bacteria. And this is after it's been infected by this guy. So what gets passed on, the yellow or the purple? They found the purple radioactive labels inside of it, and they found the yellow out in the solution. So the radioactive tags proved the thing that is being passed on is the DNA, because the purple tag was the phosphorus. All right, here's another picture, again, just to show you someone's better diagram than mine. There's a bacteriophage, so that's the virus. This one has sulfur labeled protein capsules, so the red is the sulfur tag, and you'll see the red ends up just outside, not in the cells. And then over here, phosphorus is labeling the DNA, and when they inject it, that is what's showing up, the red inside. So the phosphorus is detected inside the cell, not outside the cell, so that's truly what's being passed on to the next generation. All right, so now by this point, we've now decided, yep, DNA is the key thing. It's what we want to look at. It's what we want to study. So our next guy is Shargoff. I always think of him as like going around the farm, testing all the animals' DNA. Because you'll notice there's like chicken. Oh, this isn't the same list, but like one of the lists shows like ox and corn and chicken and cow. This one is showing a different variety. But what you'll notice in a lot of these is that the percentage of A and the percentage of T is right around the same value. A's and T's are pretty equal. So I put it right here. They tested the organisms for the amounts of A's, T's, C, and G relative to each other. A and T came out pretty equal, and C and G come out pretty equal. So he basically figured out the base pairing rule. So after that, you can easily calculate the percentage of anything. So I'll do a problem with you. If they told you that an organism was 20% A, what you know from that is that it's also 20% T, because A and T are equal. You add that up, so that's 40%. The other 60%, so 100 minus 40, I'm left with 60%. Divide that in half, 30% are C's and 30% are G's. Next, all right, Rosalind Franklin. All right, she is one of my favorites. I really am just super sympathetic to her cause, because she was a woman in 1953 who worked really hard to have her voice be heard. Being a woman in science in the 50s was a very hard place to be, and you were second guessed and you weren't always taken at your value. And it's actually super sad. She died at age 38 from cancer because all of her work was in X-ray crystallography. So actually her exposure to all of these X-rays ended up giving her ovarian cancer, and she died before she was truly recognized for the contribution she made. So she looked at X-ray crystallography studies of DNA, RNA, and proteins, and she was trying to figure out the structure of DNA based on this. And this is a very famous picture of her X-ray crystallography, and you see this X, and it's literally, if you could look down a double helix, it looks like a repeating staircase. And so she is one that really brought the understanding of double helix to the forefront. And then after she passed, they were in contact before she died. Watson and Crick did work with Rosalind, and they did come up with their idea of the model. So they are credited with the model of DNA, and that DNA structure is a double helix, and that double helix is always, okay, so the base pairing, we already know it's always A with T and C with G. But the way that it works is you always have, so let me put an A with a T and a C with a G. A's and G's are actually double rings. So you always have a double ring purine with a single ring pyrimidine. And that makes the distance of each rung or each step equal. Now out here, this is always the sugar that the base attaches to, and then between it are phosphate groups. So one nucleotide is one phosphate, one sugar, one base. So that's one T. And then the next one, phosphate, sugar, base. There's the next one. All right, Meselson and Stahl. So now we're into the 1950s. They were looking at how DNA replicates itself. So we now know that during S of interphase, DNA makes a copy of itself. And so they were studying how does it work. They had three ideas. It could be completely conservative, meaning that if this is my DNA and it makes a copy of itself, this will be my old DNA and this will be my new DNA. And they will be entirely separate. Conservative means you just use it as a blueprint, but you don't use it in your new strand. Semi-conservative would mean if this is my original, if that is my original, and I expose it to this purple, this new nucleotides, it'll be half old and half new. Or disruptive means that it doesn't really have an order to it. If that is my old strain, the new strains will look like partially blue and partially pink or purple, whatever this is. So they have to treat it with radioactive tags. You'll find that radioactive tags are super useful in biology to see where something ends up. So you use this radioactive label N15, that's the blue, so that you can follow it. They call it the heavy strain because nitrogen normally has a mass of 14 and this one has a mass of 15. So they labeled the template strand and then it ended up half in the F1 generation. And then in the next generation, it was only a quarter. And then in the next generation, it was only an eighth. So they noticed that this definitely showed that it is a semi-conservative process. So it matched what I did here. They use the strands separate, and one side is used as the template, and the new strand is built along that. Oh, there's just another, I always try and put a little visual there that's actually like done by somebody else that's much neater. And you can add these things to your Cornell notes. They make great Cornell notes. All right. Oh, and that is the end of this video. All right. Let me know if you have questions, and definitely challenge yourself. Look up, Google these experiments, see what else you can learn about them and what you can draw out according to how their experiment worked to add to your understanding of this content.